Have you ever watched a video segment or read a chapter about a particular psychiatric disorder and thought, I sometimes feel and act that way. Do I have a psychiatric disorder? This response is common for psychology students. What separates normal behavior from abnormal behavior is typically the severity, number, and duration of symptoms. To be diagnosed with a clinical psychiatric disorder, a person's symptoms must consistently interfere with thinking, feeling, mood, interpersonal relationships, or ability to function in everyday life for a prolonged period of time. As you may have guessed, the brain has a lot to do with these symptoms and behaviors. In this segment, we'll explore the underlying biological mechanisms of 3 types of psychiatric disorders: schizophrenia, depression, and anxiety. Schizophrenia is a long-term psychological disorder involving a breakdown in the relation between thought, emotion, and behavior, leading to withdrawal from reality into fantasy and delusion. The symptoms of schizophrenia are classified as positive, negative, and cognitive. Positive symptoms include hallucinations and delusions, which are in excess of typical experiences. Hearing voices, seeing things that aren't there, and feelings of paranoia or believing that people are plotting against you are all examples of positive symptoms. Clients may also experience delusions of grandeur, causing them to believe they have superpowers and that they can fly or read others' minds. Negative symptoms are behaviors and responses that seem to be missing, such as a lack of emotional and verbal responses, weak social relationships, and no motivation to succeed. Cognitive impairments include difficulty with attention, learning, memory, and rational problem solving. The causes of schizophrenia most likely involve complicated interactions between genes, environments, and experiences. Investigations of the structure and activity in the brains of people with schizophrenia has led to the development of the hypofrontality model. Hypo meaning reduced, and frontality referring to the prefrontal cortex area of the brain. As the name implies, schizophrenia is associated with decreased neural activity in the prefrontal cortex. Along with decreased neural activity, evidence has been found for structural brain differences and differences in neurotransmitter systems, most notably dopamine, glutamate, and serotonin. Underactivity in the prefrontal cortex, especially in the dopamine pathway extending from the ventral tegmental area to the prefrontal cortex, may explain the negative and cognitive symptoms of schizophrenia. Dopamine activity in the nucleus accumbens, however, which is part of the mesolimbic dopamine system, is increased. The increase in dopamine activity in the mesolimbic pathway, which extends from the ventral tegmental area to the nucleus accumbens, may underlie the hallucinations and delusions that characterize the positive symptoms of schizophrenia. The long-standing dopamine hypothesis of schizophrenia holds that the positive symptoms of schizophrenia are caused by overactivity in dopamine synapses, particularly the dopamine D2 synapses. Evidence in support of the dopamine hypothesis stems from the activity of antipsychotic medications. Dopamine is released by presynaptic neurons and binds to postsynaptic dopamine receptors. Typical antipsychotic medications bind to postsynaptic dopamine receptors and prevent dopamine from binding to and activating the receptors. The end result is decreased dopamine activity and decreased positive symptoms of schizophrenia. Recent research indicates that dopamine may not be the only neurotransmitter whose activity is altered in schizophrenia. Activity in both glutamate and serotonin neurotransmitter pathways may be altered as well. Researchers have found that the increased dopamine activity in the mesolimbic system is accompanied by decreased activity in glutamate systems. Researchers have also found that medications that antagonize or block specific serotonin receptors also increase release of dopamine in the prefrontal cortex, which improves cognitive symptoms of schizophrenia. Information about altered neural activity in specific brain regions and neurotransmitter systems is currently guiding development of new medications for treating schizophrenia. According to the National Institute of Mental Health, clinical depression is a mood disorder that affects approximately 5 to 8% of adults in America. Symptoms include extreme feelings of sadness, which may be accompanied by changes in eating and sleeping behaviors, and thoughts of suicide. While everyone experiences feelings of sadness at times, it is the long duration and severity of the symptoms that classify them as a clinical disorder. Clinical depression symptoms result in an inability to function in typical school or work settings. Current research indicates that complex interactions between genes, experiences, neurotransmitter activity, and brain structures may underlie the major mood disorders, including depression. In people with depression, researchers have found decreased activity in an area of the brain known as the subgenual anterior cingulate cortex. This region is involved with regulation of mood. Deep brain stimulation of the anterior cingulate cortex improves depressive symptoms. Activity in the amygdala, a structure involved in emotion, is increased in people with depression, which may explain the extreme feelings of sadness. Sometimes scientists develop theories about brain mechanisms based on how medications used to treat a disorder work. For many years, medications that agonize or enhance the monoamine transmitters have been used to treat the symptoms of depression. The effectiveness of these medications led to the monoamine theory of depression, which basically suggests that depression is associated with a decrease in monoamine levels. Though it has guided much research, the monoamine theory lacks empirical support and is overly simplistic. One popular class of monoamine agonists is the selective serotonin reuptake inhibitors, known as SSRIs. SSRIs block reuptake of the monoamine transmitter serotonin, resulting in an increased amount of serotonin in the synapse. The effect on receptors is immediate, but symptom relief takes several weeks. Increased levels of serotonin in the synapse cause the postsynaptic serotonin receptors to adapt and decrease in number and response to serotonin. The time course of this postsynaptic receptor downregulation corresponds with the time course for symptom relief. One area scientists are currently exploring is the link between chronic stress and clinical depression. Researchers have found that chronic stress causes cells in the hippocampus to shrink and decrease neurogenesis, the growth of new neurons. Growth of new neurons in the hippocampus is associated with learning, adaptation, and coping mechanisms. Chronic stress, therefore, is associated with disrupted learning, adaptation, and coping. Recent research suggests that decreased neurogenesis also occurs in major depressive disorder. Treatment with antidepressant medication, such as SSRIs, reverses cell shrinking in the hippocampus and promotes neuronal growth, which increases adaptation and coping, and subsequently reduces both stress and depression symptoms. Now let's take a closer look at the proposed mechanism of action of antidepressant medications like SSRIs. SSRIs induce short-term increases in serotonin levels. Chronic exposure to SSRIs results in downregulation of postsynaptic receptors, which changes second messenger systems, gene expression, and ultimately an increase in a substance known as brain-derived neurotrophic factor, or BDNF. BDNF promotes growth of new neurons and reverses the shrinkage of the hippocampus, subsequently reversing the symptoms of depression. A recent large national study of depression revealed that therapies such as cognitive behavioral therapy are as effective as antidepressant medications in reducing depression symptoms and increasing BDNF levels. Anxiety disorders are the most common mental illnesses in America, affecting about 20% of the population. Intense fear of public humiliation, irrational fear of spiders, or excessive worrying can all be symptoms of an anxiety disorder. Anxiety responses include fear symptoms, such as a pounding heart, sweating and tremors, and changes in eating and sleeping behaviors. Why our bodies have these physiological fear responses dates back to primitive times. Our autonomic nervous systems have evolved with an emergency alarm called the fight or flight response, designed to protect us against predators. When we experience anxiety, our bodies mobilize biological resources, such as blood flow, oxygen, and energy to muscles, to either fight to save ourselves or flee to safety. That same fight or flight response occurs when we experience anxiety. Instead of a real stimulus, like a predator or the feeling of a gun in your back, the stimulus, such as possible public humiliation, is imagined. In both cases, the feelings of anxiety are very real. When we perceive something as a threat to our health and safety, the emergency fight or flight response automatically kicks in. Brain regions hypothesized to play a role in anxiety disorders are the amygdala, cingulate cortex, prefrontal cortex, and insular cortex. The results of fMRI studies indicate that anxiety may be associated with increased activity in the amygdala and decreased activity in the cingulate, prefrontal, and insular cortices. Interestingly, many people suffer from both depression and anxiety, a condition known as comorbidity, or two mental illness diagnoses at once. Recent research suggests that depression and anxiety may share some common underlying neural mechanisms related to chronic stress, namely the reduction of neurogenesis and shrinking of the hippocampus, which interferes with normal adaptability mechanisms. Indirect evidence from drug treatment of anxiety disorders implies a connection between anxiety and the neurotransmitters serotonin and GABA. Medications that agonize serotonin and GABA reduce the symptoms of anxiety. A decrease in serotonin's inhibition of the amygdala, basal ganglia, and cortex, and a decrease in GABA's inhibition of the cortex may play a role in anxiety. Both GABA agonist drugs and serotonin agonist drugs like SSRIs effectively treat the symptoms of anxiety. You may recall from the previous segment that SSRIs are also effective in treating depression. If both anxiety and depression share similar underlying brain processes that are related to chronic stress, then it's no surprise that the same treatments would relieve the symptoms of anxiety and depression.